Okay, so we left off, well, we had a test on Monday, but we left off sort of in our lipids chapter talking about our next major class of lipids, which I think is, let's see, 1, 2, 3 probably, but we'll just not put a number on it. So steroids and cholesterol. So the part we want, or the part that'll let us recognize a steroid, is they all have in common that cholesterol structure. Yes. And the cholesterol structure is a bunch of 6-membered rings joined together and then one 5-membered ring on it. So it's a bunch of hexoses, kind of, except it's not sugar, so it doesn't really count. Oops, let's start that over. Leave a little bit more room. It's drawn on page 755 if you want to look in your book, 775. We're just redrawing it here because we want to have practice doing it. To me, it kind of always reminds me of a honeycomb. So I've got a 6-membered ring, 6-membered ring, 6-membered ring, 5-membered ring, and technically they also count all of the carbons. I won't hold you responsible for it. We won't say what functional group is on this numbered carbon because I don't want you to memorize the numbering scheme for something like this. But, for instance, it would be a perfectly legitimate question to say, what is the functional group on this molecule, or what makes it special? Because we're not going to memorize the structure of any steroids. If I ask you a question about a steroid, it's going to be either what structural features are on there, or to identify the molecule or the class of molecule by recognizing the cholesterol structure or core, and the functions of cholesterols. Just like I'm not making you memorize any fatty acids or any specific molecules, but you have to know how to put them together to make a larger entity like a phospholipid or something like that. So the thing they share in common is that cholesterol structure, but they're highly diverse in function. And so they can be used in membrane components. So one of the things that's kind of kept shoved towards the end of this chapter is, cellular membranes. We've already talked about liposomes and micelles. Those are just made of fatty acid molecules and or phospholipid type molecules. This would be something that would be incorporated in a membrane, not just by itself, but in conjunction with lots of other things. And what it really does is it gives it rigidity. What? Oh, I can make sure there. Tell me if it flickers some more, because frankly I'm not looking at the screen. I guess I could write like this. That'd be even a bigger skill, better skill, wouldn't it? Like to be able to write without seeing what you're writing, kind of. I think that would be tough on the brain. So anyway, that membrane section is saved till the last part of this chapter, and I'll probably let you guys read it on your own since it's nothing beyond, a basic high school biology level discussion of it. We have things like bile salts. So those of you in nutrition, what are bile salts used for? So they aid digestion of fat. We won't specifically, I don't know why it's flickering then. Maybe it's the way I'm pressing down on the screen, but that doesn't seem to do it either. I don't know what's going on. We'll take a look at it here in a minute if it still keeps flickering. We won't look specifically at the role of bile salts in the digestion of fats, but we will look at how fats are digested and how we extract the energy from them, because we'll be looking at beta oxidation, which I know Todd covered kind of in class, but not really. It was like an extra credit assignment. So we'll go through that in more detail. There's also, let's see, are these there's like ergosterol, which is what is the precursor to vitamin D, which is an important vitamin. There's, it can have so sometimes it flickers when I write, sometimes it doesn't. I don't get it. It doesn't really matter. Doesn't seem to matter how fast I write. Well, maybe if I don't know. It's just going to have to be what it is. We only have to put up with it for another rest of this semester, and then supposedly we have brand new technology in the new building, which doesn't help you guys necessarily, but I'm not going to worry about it too much, which is a heart medication or heart drug. It's also responsible for any of the, oh boy, that is kind of annoying, isn't it? Adrenal, coronary. Yeah, it's going to cause an epileptic fit here in a minute, I think. Let's see. Check all the cords. That's plugged in pretty good, isn't it? Yes. Yes. I don't think it's on that end. Otherwise, it would flicker when I'm not doing anything, but it seems to only really flicker if I'm touching something. Which play important roles in metabolism. Maybe it is how I'm resting my hand on there. I'll try not that. And probably the one we're most familiar with for steroids and things like that is they're responsible for the sex hormones, which is responsible for sexual characteristics, and reproduction. Boy, that's really kind of annoying, but we'll just have to deal with it. One of the things that we also want to point out about cholesterol that we've kind of looked at before, so kind of draw an analogy, is cholesterol is sort of the starting component for lots of molecules. For a series of molecules. What was another example of a specific molecule that we've looked at in this chapter? It was the starting molecule for a series. Which one, Jeff? Apparently this room is far too hot also, and it's overheating the projector. So there's all sorts of fun and wacky things going wrong. Maybe we should just write on the right whiteboard, but... Well, I think it really is, I think it might be that, but I think it's also a partial heat thing. Like, if you go stand on the desk, you can literally feel the temperature in the room rise as you go up. So the heating in this building is not terribly good either, or the air circulation. So what was the prior example that we looked at of something that starts off a molecular or a biological pathway, or a series of compounds that are made from a starting compound? Excellent test question. We'll leave that for the test then. So if you look on page 776, figure 28.5, what we see is that we can take the structure of cholesterol and we can modify it, and we get progesterone. Now, I don't expect you to memorize the chemical changes that are occurring. I don't expect you to memorize that cholesterol leads specifically to progesterone. What I do expect you to understand is the concept of the pathways, and that there are certain molecules in your body that are sort of the starting point for a large series of other molecules. It's kind of like this is a building block or a 2x4 that you're building your house out of. It's used in a lot of places. And progesterone is a female sex hormone, right? I guess we can be cheap. We'll go like this. There we go. And what I kind of find interesting is progesterone is turned into testosterone, which is, of course, the male sex hormone. But that is also then turned into estradiol, which is another female sex hormone. And so there's a whole series of chemical reactions that are going on to make these hormones. And you know, if any of that process is screwed up, we of course get or have problems. And controlling these pathways is, you know, part of, oh gosh, what's the word I'm looking for? Birth control type things, where you know you might control which of these hormones are expressed and when they're expressed and things like that. There's also a whole bunch of other things that cholesterol can be made into. One of them, for instance, is, let's see, like cholic acid. That's an example of a bile salt. So really, cholesterol is kind of the root of those. It makes ergosteroid. We mentioned that one too. That's the precursor that eventually leads to vitamin D. It also makes some compounds called cytostanol esters. And we'll talk about that later. What this actually leads to is it blocks absorption of cholesterol. So it's kind of funny, they make a molecule that actually blocks the absorption of its precursor. So if you make a bunch of cytostanol ester, then you'll actually have less cholesterol absorbed in your blood or in the intestines. And so there's all sorts of things that the steroids or that cholesterol core gets turned into. And so we want to realize that when we're looking in biology, there's always sort of key molecules that are reused kind of obsessively. For instance, in the carbohydrate chapter, what single molecule is the most used? Yeah, glucose. So glucose is the most common monosaccharide. Every disaccharide that's important has a glucose in it, right? And all of the various forms of the polysaccharides that we studied, the starch, cellulose, and all those, are all just different forms of glucose, or glucose connected by you know, different glycosidic bonds. And so glucose is like a fundamental building block of your body too. It's an example of that. Same thing we'll learn with, you know, when we go through the lipid biological pathways, you'll notice that there are certain molecules that are used in multiple pathways that link them together, and so are more important than, you know, say a miscellaneous one. So that kind of ends our discussion of the molecules, the different types of lipids that we're going to have on the test. We can look at their uses too, and we've already kind of got there. So this would be biological applications, maybe we'll call it. And I've done some of it out of order because, you know, I covered, for instance, an example of that is micelles and liposomes. We covered that when we sort of covered fatty acids and the hydrophobic, hydrophilic nature of those. And so, for instance, that's an example of something that you can go and read, or, you know, that we've already covered. We're going to talk right now about atherosclerosis, which I really dislike that word. It's very much a mouthful to say. It's also very much a mouthful to spell, so that's probably the only time you're going to see me write the word atherosclerosis, because I'm not sure I could ever write it again or spell it correctly if I wanted to. Does anyone know what atherosclerosis is? And I guess I'll put the last one. So this is going to be just kind of a list, of course, membranes. And so you can read this on your own. This is already done. And this is what we're doing. So we'll have our entire page for atherosclerosis. Atherosclerosis. There we go. That's my shorthand for it. ASATH ... Or maybe Jay shouldn't be lazy. Maybe Jay's just in a weird mood today. So atherosclerosis. So does anyone know what that is? It's something I would guess at least one person's parents suffer from in this room, and certainly some family member or friend you'll know has suffered from this disease. What is it? So it's the, what? Scoliosis. What? So it's a metabolic disease. So what does it mean when we say it's a metabolic disease? So it's certainly involved with metabolism, right? And normally when we say something's like a metabolic disease, I think it's a misnomer, because I usually, when I think of the word disease, I think of things caused by bacteria and viruses, right? I would say it's more like a metabolic misfunction, meaning your body, a pathway in your body is disrupted or doesn't function, or you overload the pathway by basically eating too many lipids and things like that. And what it is it's the accumulation of lipids in blood vessels. And that can lead to heart attacks, high blood pressure, and blood clots. And maybe I'm kind of going a little out of order. I mean, arguably I should say that high blood pressure comes before heart attacks, because normally heart attacks are caused by high blood pressure, right? And so what happens is your body fails to properly transport, store, and dispose of lipids. Oops, maybe it is my computer, because I think I just crashed it. Let's see, can I go to the next slide? Okay, maybe it's working now. I think it's something to do with this computer that's causing the flickering. And so what happens then is, because you fail to properly transport, store, or dispose of the lipids in your body, they tend to accumulate on the artery walls. I will just not rest my hand on there as much as possible, but that doesn't necessarily seem to be helping terribly well either. So I don't know what the way it is. So they accumulate, they are oxidized, and then they're scavenged by white blood cells. You guys are all familiar with WBC as an abbreviation for white blood cell. I would imagine Sarah uses that one a lot. And that tends to lead to them to be bloated and to both hardening and narrowing of arteries. Okay, maybe I'll unplug that. That's could be causing it. Could just be that this connector is flaky, but if I wiggle the connector, it doesn't cause anything. Let's see, if I do this, then I broke it totally? No. Just kidding, we'll fix it. Oops, upside down, right side up. There we go. Let's see if that helped fix it or not. I don't know if there's something under there or what the deal is. There's a loose screw here. What? Which? Oh, the white blood cells become bloated, meaning they kind of make this foaminess and then they get stuck in the arteries. And so that's what specifically leads to the hardening and narrowing of the arteries, is what leads to the high blood pressure, and that eventually leads to heart attacks. Or if it fragments, meaning a small piece of that bloated foam or that hard coronary hardening breaks off, that can also lead to then the blood clots. So how many people have had a parent have a heart attack? Or grandparents, I guess, too. You guys are all pretty darn young. It might not be quite your parents' time yet. Yeah. Or how many people know that their parents or grandparents are on high blood medicine, blood medications? Yeah, my dad's on like 5 different ones and stuff like that. He hasn't had a heart attack, but they kind of want him to watch all that sort of stuff. So I would say this, along with cancer, if we talk about that in the DNA chapter, are probably the 2 single largest factors or causes of death in the United States. So these things are important. It's good to have a basic idea of what's going on. So what this does is, we talked about, or I kind of mentioned it, is basically improper transport of lipids. So what we want to do is we want to look at that transport scheme for lipids. And so there's a bunch of different types of transporters for lipids, I guess we'd call it, and then there's sort of a pathway, and it starts in the liver, and the liver produces what are called VLDLs, because no one wants to write out the whole word, but I'll write it out. VLDL stands for very low density lipoproteins. And this is what actually transports the lipids. And if you want, there's a nice picture of it in figure 28.8. It's basically a micelle plus a bunch of proteins that help to package it, help it to pass through cell walls and things like that. And what this is used to do is this transports, or the VLDLs transport lipids to adipose tissue. Adipose tissue. You guys know what adipose tissue is, basically? From Sarah's class? Yes, no? Basically fat. In fact, I saw a very interesting picture. I should have brought it in. It's kind of disgusting. They showed a person holding up 5 pounds of fat versus 5 pounds of muscle, and 5 pounds of fat has about 5 times the volume of 5 pounds of muscle. So, you know, if someone's like small and muscly, you know, they can actually weigh more than you might think, simply because muscle weighs a lot. And so if you can convert, you know, fat to muscle, you might not change any weight. In fact, I've heard that sometimes when you start working out, you actually start to gain weight because, you know, you're changing the fat to muscle. And so you're not really losing it, but you will be at the same time losing volume, obviously. So that's responsible for the transportation to the lipids. And at some point in that chain, the VLDLs are converted to LDLs. Which stands for, of course, low density lipoproteins. And these are what transport to peripheral tissue for energy. So they transport lipids to peripheral tissue. I don't even know how to spell that word. Peripheral tissue. Yes, we will actually make that distinction here in a second. These are considered bad, meaning having an excess of them is bad. You certainly need a certain amount of them. And that's the other thing that's always tough with anything in biology, and really probably with chemistry too, since biology is just really complex chemistry, is that low quantities are that, you know, there's always a sweet spot for something. There's always the maximum efficiency point. If you have too few or too much of anything, it's generally bad. So these are considered bad for you, especially if because they're kind of faulty, meaning they're a poor mechanism for transporting lipoproteins. And so the more of them you have, the more lipoproteins you're trying to transport, and the more lipoproteins that kind of escape that. I like to think about it as you've got a box full of like cats, and you're transporting them someplace, and the cats get out and run around. And so, you know, if you have a very good cat transport system, you have pretty good blood pressure and things like that. You know, you don't have a lot of lipoproteins free and hardening in your arteries, but if you let too many cats get loose, it's just hell. I don't know if that's a good analogy or not. Anyway, they transport the lipids to the peripheral tissue for energy. And so, I mean, it's a system that we need. We need to be able to transport these lipids to different cells in the body to be used as energy. It's just that we need them to be transported properly, not kind of faultily. And then the other part of this, or the last kind of part of this mechanism, is, of course, HDLs, which, can you guess what they're going to stand for? Yeah, high density lipoproteins. And they're basically scavengers. What they're responsible for is returning cholesterol and lipids to the liver. So if we were kind of sticking with our analogy, HDLs would kind of be like the person that's responsible for catching all the stray cats that escape and returning them to liver to be, repackaged or properly disposed of. And so these are generally considered good for you, meaning that if you have a high HDL, you've got a very good ability to scavenge any of them in there. And what it really comes down to is not so much that one is good and one is bad, but it's the ratio of LDL to HDL. And that what most studies show is that if I have a high levels or a high ratio of LDL, then that leads to increased risks of heart attacks or atherosclerosis, I guess we could say it that way, sclerosis. And high levels or a large ratio favoring HDLs has generally been proven to decrease the risk of atherosclerosis. Or conversely, if I have very low levels of HDL, that also increases the risk, right? And so, in general, it's important to have the proper ratio. If you have, good LDL levels but really poor HDL levels, then you're still going to be at risk. And if you have super high HDL levels, you can actually have fairly high LDL levels too. So it's the ratio of them that we're kind of looking at to do it. And so, for instance, a low cholesterol diet isn't necessarily enough to, combat high blood pressure and atherosclerosis if you're still getting the wrong ratio of LDLs and HDLs in your diet. So, treatments for this disease focus in on these metabolic pathways. And this is really the main point that we want to talk about is, we've got these pathways established. If we study up on how they work, meaning we do research on them, then we can design drugs that specifically affect these metabolic pathways and help us to be better people. I used to be of the opinion, and some days I'm kind of going back towards that the only medication you should ever take is aspirin, and even that sparingly, meaning I was way anti-drug. As a kid, or even, as an adult, I don't tend to take much stuff. But I guess as I've gotten older, I've realized that probably some of it's good. But, to me, the first place you always want to start at is diet and exercise. And so the first treatment for this is that if you have a proper diet, and when we say diet, we don't mean that, you're dieting to lose weight. We're saying just your general, what? Well, it would mean that if you are eating the cafeteria food, you're probably trying to focus more on fruits, the vegetables, and salads. Is it all canned fruit these days? Because I always saw that there was oranges and apples and bananas sitting in the corner. Yeah, I understand it's not exactly easy. Of course, I'll argue that it's not even easy in Rangeley to get fresh produce. I mean, really, I feel like I should make a trip to Vernal a week to pick up fresh fruits and stuff. So diet and exercise is probably the first treatment stage to it. And then the rest are sort of looking at drugs. So the idea is that you want to both lower cholesterol by a variety of ways. And so one way that you can do it is you can decrease triacylglycerides. by the existing levels. So you can basically take triacylglycerides that already exist and destroy them. Now, I don't expect you to know the class of drugs. I don't expect you to say fibrate drugs do this. What I expect you to do is say, how are ways that we can combat atherosclerosis? And one of the ways is to decrease the existing levels of triacylglycerides. Meaning, that's the key. What specific drug does it? I don't care so much. I mean, it's certainly a legitimate question that I could put in. I'm just telling you I won't. What I really want to, or why I put the drugs names up there is, you should write them all down, and when you go home next weekend or the weekend after that, or you call your parents, ask them how many of these drugs they've heard of and or are taking currently. And I would guess that most people's parents and or grandparents, they'll know someone that's taking one of these drugs. Another way to do it is to decrease the metabolic synthesis. And some of these drugs can decrease it by up to 80%. So what that says is that even though you eat it, your body doesn't metabolize it necessarily and turn it into or build cholesterol out of it. And those are called the statin drugs. And the brand names for those are Mevacor, Lipitor, and Zocor. And for instance, I definitely know my dad's on one of those two. I don't remember. I was on one and then they switched him over to the other one, and I don't really keep track of, my dad's medicine cabinet. This is kind of one of those ones that I'm kind of like, ugh, I don't know if it works. But you can increase the excretion. And it does that by binding to the bile acids and basically stopping the absorption. So one way that we can have less cholesterol is if we basically shit it all out, meaning we don't absorb it in our body, we just get rid of it. And that's called the Resin Drugs. And brand names, bile acids. So we said that bile acids are important for the digestion of fats. So if we bind up those bile acids and we prevent the digestion of fats, we actually increase the excretion of them, stopping absorption. So that is the resin drugs like Cholestid and Questran. And really, that has the same effect as making sure you have a high fiber diet. So if your diet is low in fiber, the drugs will help, or you can simply have a fat or a high fiber diet. The last one that we'll take a look at is we can decrease intestinal absorption. I'm not sure that not resting my hand on the computer is actually helping something. I know if nothing else, it makes my handwriting really crappy if you can't rest your hand. Try to imagine writing like this without resting your hand on the desk. I imagine it's a pain in the butt. And the way they're doing this is they use plant sterols. So they actually use a steroid to do this. Sterols. And we mentioned one of them, that cytostanol. And where you can find that is in Benicol, which is basically just a butter substitute. And also the other one I said was cytosterol. And again, don't memorize the brand names, memorize kind of what's happening or understand the mechanisms that we're trying to affect. That's found in Take Control. So that's another butter substitute. So if you take these butters that have these compounds already or added to them, then you can decrease intestinal absorption by, like, say, 10 to 20%. So, you could accomplish the same thing by simply changing your diet and having less of it in your diet. But if you're not willing to change things like that, and you are willing to, say, for instance, use this butter with these added compounds in it, then you can cut down on it that way too. But to me, really, the beginning place is probably diet. Now, I don't understand enough about the disease to know, but to know all the specifics, but certainly there are genetic risk factors. Some people have a genetic history and, or of high blood pressure, or, basically what that's saying is that some metabolic pathway in their body isn't super well regulated, or isn't as well regulated and well controlled as it can be or should be. So that's atherosclerosis, and that's an example of a metabolic use of lipids, or the control of them, I guess, and then ways that we can kind of change or affect those. And one of the things that we should learn is that, the way your book kind of goes through the biochemistry is it shows you all the chemistry parts of it. It shows you what the molecules look like, the functional groups on them, some of their basic physical properties, and then it likes to give you a couple examples of how that affects things, right? So for like carbohydrates, what we focused in on was differences between starch and cellulose, right? We brought up amylose, amylopectin, and what's the other one? Glycogen. We didn't cover, for instance, blood grouping, and maybe I should have added that into the lecture. We kind of ran out of time anyway. And so, like blood typing is another example of a good use for it. Or if you're reading your chapter, one of the things that is big is, of course, sugar substitutes, right? So one way you can cut down on calories, one way you can cut down on your intake like that is to simply use, for the sweetness flavor in things, sugar substitutes. And so, again, your book likes to present a couple examples of uses. We generally will lecture or cover a couple of them and then leave one or two for you guys to read on your own. So I think the only thing I really haven't covered is something that we're going to cover in lab, but just so that it's for the completeness sakes, I'm going to put it there. And that's some reactions that we're going to look at in lab. And so there's three main reactions that we want to talk about. I'm not going to give an example of, for instance, minus H2O and plus H2O reactions. That's simply how we put together the molecules and how we've taken them apart, meaning we've already covered that elsewhere. So I'm not going to put an example in this reaction. The second one that we kind of want to talk about is the concept of hydrogenation and dehydrogenation. And I probably should have just thrown these in the notes under our section on cis, trans, and saturated and unsaturated fats. But this is a reaction we've already kind of looked at. For instance, if I have a gigantic molecule, or carboxylic acid, what was that yeah, that's our reduction, right? And we can use reduction reactions to do what? So, oh, I'm sorry, I forgot to do that. Have to have the double bond in there, otherwise the reaction isn't going to really go. What was, you gain bonds to hydrogen, right? And so this was our reaction where we add a hydrogen to one side of the double bond and we add a hydrogen to the other side. Now, I'm not being exact in how big I'm making this molecule, so don't worry about how many carbons are on each side. Focus on the fact that there's a carbon-carbon double bond and not a carbon bond, and just carbon-carbon single bonds on the other end of it. And that, of course, would be an example of a hydrogenation reaction. So we're taking a unsaturated fatty acid, and making a saturated fatty acid. Now, notice that I sometimes call them carboxylic acids and I sometimes call them fatty acids. That's because most of the time I'm a chemist, but we're supposed to be doing biochemistry here, so realize that I could have also written unsaturated carboxylic acids to saturated carboxylic acids, and that's the same thing, isn't it? So kind of get used to that idea that fatty acid and carboxylic acid are just the same names for things. And what type of reaction is this also? It's a reduction, and it's also a addition reaction. Does that one ring a bell from our chemistry chapters? Seems like I had a couple people poking their heads up and going, oh yeah, I remember that one. And then of course dehydrogenation is the reverse process. So if we go this way, meaning we have to remove hydrogen. And so if we want to give an example of that reaction, we would start off with a molecule like this. We would remove hydrogen from the molecule and introduce a carbon-carbon double bond someplace in it. The location is irrelevant. Irrelevant, meaning I don't care where you show the double bond on that molecule. And we should also realize that it really leads to both cis and trans isomers. Even though, for instance, we won't necessarily draw it showing two products, we will, of course, say that, you know, we realize that anytime we put a carbon-carbon double bond in a molecule, we almost invariably create a cis-trans isomer. And so one of the reasons that unsaturated fatty acids that are artificial are bad is because they tend to make a lot of the trans isomer. So while in general unsaturated fatty acids are good, meaning having that double bond in there is useful and normal, having that double bond in there and having it be the trans isomer, which is what you would produce if you do it chemically, as opposed to, you know, having a biological mechanism for doing it in plants that produces mostly a cis isomer. So this is where you see foods, you know, it's listed, this food contains trans fatty acids. Of course, these days what I really notice is they like to just list foods that contain, or they say does not contain any trans fatty acids, right? But I love how they put that sticker on things like a banana. I'm pretty sure a banana never contained trans fatty acids in the first place, yet, you know, they'll put it there. Or a lot of other products seem to be slapping on that no trans fats in this product. Well, they never contained any trans fats to begin with, I don't think. It's not like they removed them from the product. So I always get a laugh out of looking at some things and going like, wow, it's trans fat free. How novel, when it never contained them in the first place. So that's about it for lipids. So that means that we're transitioning to chapter 29 on amino acids. That's a 9. Maybe it was 27, I don't know. I looked down, maybe I was thinking the wrong chapter. I have no idea. So this would be considered our last major class of compounds. I guess really technically the chapter is probably called amino acids and proteins if they're really smart, because what are proteins made out of? Yeah, so really if I look in the book it's called amino acids, polypeptides, and proteins. Amino acids are the starting point. One of the things I like to do is make analogies. I think an amino acid is kind of like a fatty acid, is kind of like a mono or carbohydrate, or I guess we should say specifically a monosaccharide, meaning that's the fundamental building block. Now, the tough part about protein chemistry is that how many amino acids are there? 20. 20 common ones, 20 ones that basically in humans make up probably like 99.999% of everything. How many different types of monosaccharides are there that are important? 3. Technically, I'm going to say there's really one that's vastly used, meaning glucose is by far the most important one. The other ones are important, don't get me wrong. I'm just saying that, you know, in that case, glucose is really that. And how many fatty acids are there? There's a bunch. I don't think anyone ever counts them, really. So amino acids are going to be a little bit more complicated, or at least the chemistry involved is much more complicated because there's much more of them. And then we're also going to talk about polypeptides. I don't know if there's a good example for fatty acids, but I would say that this is our same thing as our phospholipids, meaning it's the next step up in it. And for sugars, I would say that this is more like our disaccharids. And so what a polypeptide simply is it's a very short amino acid chain. So it's on the order of, I don't remember off the top of my head if I even have it in my notes, nor do I really truly care what the number is. Mostly the fact that it's a short or small number. I don't even see it in my notes. We're talking, 20 to 30 amino acids long at most. And let's see, do I even have, you would think I would have had that somewhere in there, but like I said, I don't care about the number. I just want you to know that small fragments are called polypeptides. And then when we start talking about proteins, that's kind of like our lipid bilayers, meaning they're very complex. Or that's like talking about starch and cellulose. Here we're talking that this might be made up of thousands of amino acids. And so really, amino acids to polypeptides to proteins are just getting bigger and bigger in size scales. So there's a variety of properties that they have, or maybe I should say a variety of uses, properties and uses, and they are much more varied to my mind than definitely fatty acids, which really the only thing we talk about is lipid bilayers and as an energy source, and they're far more versatile than, say, glucose and carbohydrates, even though carbohydrates can make the difference between starch and cellulose, which is a pretty large difference. So, for instance, they're used to transport blood. They're used to make enzymes. And enzymes basically speed up chemical reactions. We'll talk about the structure of enzymes in this chapter, but we'll talk about their functioning in chapter 30. They make up a lot of structural components. Whereas, cellulose is about the only major structural component of carbohydrates. Here, this thing, these can make things as varied as spider silk, feather, hair, hooves, silk. S-I-L-K. You've heard of silk before? I think one of the coolest ones, I call it fish antifreeze. There's a protein that fish in very northern waters, we're talking Antarctic waters, all have, that essentially keep their blood from freezing. So it's kind of like fish antifreeze. Their nutrition or energy. And they're responsible primarily for both growth and maintenance. But they're also a source of energy, along with fats and carbs or carbohydrates. Let's see, what else do we want to say? Oh, sometimes the individual amino acids have uses. So there's a few amino acids that just the molecule itself has a use, and it's not necessarily always built into a building block. For instance, glycine, glutamine, and tyrosine, tyrosine, are all neurotransmitters. Tyrosine, for instance, is the precursor or the parent molecule for adrenaline. So tyrosine leads to adrenaline. And for instance, you guys are probably familiar with histamine. What's histamine the precursor for a lot of? It's part of your immune response. And so it's responsible for the symptoms associated with like colds and things like that and inflammation. So a lot of the individual amino acids have very specific uses. So here's a laundry list of why proteins are important. What you have to do for a test, just like with carbohydrates, and I said you don't have to memorize, for instance, the 20 amino acids, just like you didn't have to memorize the structure of the carbohydrates, but do remember where you find them in nature and where or some important uses for them. So have an example or two in mind, but don't memorize the entire laundry list. We can label proteins as being essential. What does essential proteins mean? Or I'm sorry, not proteins, amino acids. There's 10 of them. Yeah, need to be gotten in diet because your body cannot produce. One of the things that, for instance, if you're a vegan or vegetarian, that you do have to worry about is making sure you get all 10 of those essential amino acids. And so quite often you have to make sure that you eat enough like beans and nuts and things like that to contribute it. If you have any sort of meat in your diet, you pretty much are guaranteed to get all of those amino acids in there. And so we can also label food as being complete foods versus incomplete foods. And that complete or not simply means that it contains all of the essential amino acids versus it does not contain all the amino acids. And so in general, if it's from an animal, most of those proteins are considered complete. Animal protein is a good example of this. And mostly plant and vegetable proteins are incomplete, meaning they don't contain all the essential amino acids, meaning there are examples of vegetable and plant proteins that are complete, but you have to eat very specific ones and in specific quantities. So maybe I should start, since this is a chemistry class, we probably should have started with what the structure of an amino acid is. And actually we'll put this as under general structure. So what do you think functional groups are contained in an amino acid? The name itself kind of gives it away. Amine groups and carboxylic acid groups. So the generalized structure for this, and depending upon the book you read or how you're looking at it, they'll draw these in different ways. I like to draw it this way for a specific reason, so that when we start doing reactions with them, they're oriented the right way. But you will definitely see these written in different ways. So things that things that amino acids all have in common is, of course, the carboxylic acid group. They all have an amine group. And then this is varies. So the R group varies for each amino acid. And that's what makes each amino acid unique, is they have a variety of different R groups. That R group can be as simple as another hydrogen atom, or it can be as complex as benzene rings and various other more complex shapes. The carbon that kind of connects it all together is quite often called the alpha carbon. What else do you notice about these molecules? It's got a carbon atom with how many different things attached? 4. So most amino acids are chiral. Now when I say most, obviously if I've got 2 hydrogens on that carbon, then that amino acid is not chiral, right? Or if I have a second carboxylic acid or an amine group in that spot, then that molecule is not chiral. So most, meaning I think 19 out of 20 amino acids are chiral. And so if we kind of draw them like chiral molecules, then we should put the carboxylic acid functional group at the top. We put the hydrogen, and we use the amine group to determine whether it's one or the other, and then we stick the R group facing down. So if we're thinking about it kind of as a chiral molecule, we tend to draw it this way. And this would be then the D isomer. And if I draw it with the NH2 on the left side, that of course makes it the L isomer. Now, in general, we were very specific to always say alpha D-glucose and various things like that. We tend to not worry about that too much for when we're talking about amino acids, because really we only use almost exclusively the L amino acids. So the L isomer is about 100% of the amino acids. There are a few naturally occurring D variants. So I think it's kind of funny. We use D carbohydrates, but we use L amino acids. I don't know if there's a reason behind that, if it gives us some special feature, if it just happens to be that at some point when they first started using amino acids, or when whatever the primordial ooze that life crawled out of, or however it evolved or was created, who knows, they had to make a choice and use one or the other, right? Because it makes no sense. It doesn't make any sense to have both used, because then you have to have 2 sets of mechanisms for everything. Instead, you want to use only one type specifically. And I don't know why that was, I don't know why it's L for one and D for the other. But we basically exclusively use the L amino acids. So let's take a look at those amino acid side chains. There's a variety of different ways that we can kind of divide them up. The major scheme is there's a list of nonpolar side chains. We can have the polar side chains. We can have ones that are considered acidic. And we can have our basic side chains. And so in some books, and I don't believe your book does it. No, your book, that was one of the changes. That was actually, your book used to just list all 20 amino acids in alphabetical order. Now your book lists all 20 amino acids and puts them into these groupings or categories. So what would make something nonpolar? What type of side chains are considered nonpolar? If you had your book with you, I'd say go look on page 796, and the answer is right there. But you guys know enough chemistry to tell me what types of things are nonpolar. Things that aren't charged, things that don't have separations of charges built into them. They what? What?
There is no point. When it's like all of them like pushed together so it doesn't like glue.
Not a clue.
That's polar, not polar.
Oh, I see what you're saying. So
Yeah,
if we have a dipole in it, then the molecule itself has a asymmetry in the charge, right? And we can think about that as pushing and pulling. I see what you're saying. Yes. So what type of functional groups are nonpolar? This is straight up from chapters 19 through 26. What? Well, hydrogen really isn't a functional group. It's an atom, right?
So like we're talking about like amines and stuff.
Yeah, and stuff. So let's just start. Amines are polar, right? So nonpolar groups are basically anything that is aliphatic, which simply means that it's carbon-carbon single bonds, double bonds, or triple bonds. And or aromatic, meaning it contains a benzene ring in it. So basically, if it's just carbon and hydrogen in the side chain, then we would consider it mostly non-functional. And the reason we divide these up is because these are hydrophobic. And so we're going to come back and we're going to understand that if I have a protein that has large sections of the protein that are mostly non-polar side chains, that tends to be very hydrophobic, and that tends to be curled up on the inside of the protein away from water. What type of side chains are polar? Wow, if we gave the organic chapter test again, I do not think it would go as well. Think about things that cause things to have asymmetric distributions of charge, meaning partial positives and partial negatives. What do we always draw those on? Yeah, things that can make hydrogen bonds. So how about OH groups or alcohols? What other types of things were considered polar? Yeah, those are but that's actually generally considered down here.
Is it nitrogen?
And let me point out that when we say carboxylic acid groups make amino acids acidic, we're not talking about this carboxylic acid group, because every amino acid has that one, right? We're talking about R groups, right? I had someone write down that all amino acids are acidic ones, and so I try to stop that. Just like, for instance, things that are basic are amines. And again, we mean extra. And so really, when we're looking at polar compounds, it's going to be alcohols, amides, and alcohol groups. And you're right. I mean, carboxylic acids and amines are going to be polar, but they're labeled or stuck under the acidic and the basic parts because that's the more important property. So things that are polar tend to be, of course, hydrophilic. And then things that are acidic and basic are also both hydrophilic. But we're going to learn more importantly that these are responsible for forming what we call salt bridges. And they can also form ionic bonds really well. And so acidic and basic functional groups will interact with each other too to form ionic bonds or salt bridges. So they can interact with each other. And so what's important about an amino acid is not that it has an amine group, not that it has a carboxylic acid group, but what really gives each amino acid its individual character and its individual usefulness is what it has on there for a side chain. And so while everyone has those two functional groups, what we really want to focus our attention to is the various side chains. And I think we should probably stop right there. It's an okay breaking point. Let us make homework 28. A and B due Friday. And you can also start in on 29A. You could probably do at least half. Meaning, you can put it off and do it, on Monday or next week or something when you've forgotten some of the material, or you can start to think about putting it here. And then tomorrow in lab, we'll get a start in on the lab on lipids. And I would also suggest bring your books. Lab, class, and office hours.